Okay, so now this is the second part of, we're going to go into organelles and what they do and how they relate to biotechnology. Okay, so here's structure of cells. Okay, and I had you do that assignment at the Utah Genetics website. So it goes into briefly what each of the organelles do. And for right now, that's all we really need to cover. Let's see, here's a plant cell. So things that make it different is it has chloroplasts, okay, which are going to use energy from the sun to make chemical energy. There's the vacuole. Okay, it also has a cell wall. Okay, animal cells, no cell wall. Okay, they have cell membrane. Okay, plant cells still have a cell membrane. It's just inside of there. Mitochondria both have. Lysosomes both have. Golgi apparatus both would have. Okay, so we're just going to keep moving along. Okay, so cell walls are found in plants. They can also be found in fungi. Okay, so fungi, I mean like mushrooms. They're microscopic fungi that you don't necessarily see. Like anytime you have something fuzzy growing in your fridge, that's a fungus. So they're outside the plasma membrane. They provide structure. Okay, so plant, bacteria, fungal cells. Okay, not in animal cells. Okay, so here's a. Okay, so I'm not going to make you memorize everything on this picture. That would be that's not the point. The idea is these are bacterial. These two are bacterial cells. So here is cell wall is the brown, okay, and then this yellow is the plasma membrane that's inside. Okay, and then out here in the gram-negative bacteria, so there's two different kinds of bacteria, and we categorize them by what's found in the cell wall. So gram-positive have this thick peptidoglycan layer, whereas gram-negative have a lipopolysaccharide layer. Okay, and so here's the membrane. That's what I want you to get. Okay, here's plant cell wall. Okay, so out here, the big thing about it is it's got cellulose in it. So cellulose is a carbohydrate that we can't break down. So whenever you talk about having a high fiber diet, someone has a high fiber diet, cellulose is what's going through them and cleaning you out, making you poop. Okay, and then this one is this is chitin, and chitin's found in fungi. Okay, so out here, this is the cell wall. And then in here, this is the plasma membrane. So let me see if I can make my pencil. Okay, so plasma membrane in here, plasma membrane in here, plasma membrane, plasma membrane. Okay. Okay, so now we'll work our way in. Okay, so the plasma membrane, the plasma membrane, what it's going to do, okay, is it's going to regulate the movement of things in and out of the cell. Okay, so it's made out of phospholipids and proteins that are embedded into it. Okay, so there's different proteins in here and they have different functions. Okay, so they could serve as molecules for transport. Okay, they could serve as enzymes. Okay, and we'll talk about enzymes. They could serve as kind of like an ID tag. Okay, which I think that's really interesting. And they could be structure, like they could provide some like skeletal support. Okay, so here's a picture. Okay, so the first thing I want you to see is this phospholipid bilayer. Okay, so the phospholipid bilayer, and it's written out here. Okay, what it's made out of is it's made out of a head. Okay, and the head is actually polar. Okay, and hydrophilic, so it's water loving. Okay, so it's out here. Okay, and then it's got these fatty acid tails. Okay, and the fatty acid tails are nonpolar or hydrophobic. They don't want to be around water. Okay, and then when we say it's got a phospholipid bilayer, it goes like this. So it's got two layers of these. So you can see here's a row of them, then here's another row of them. So here's the head, the phosphate head that likes water, and then the lipid tail, which says no water. Okay, so the lipid tails go together because they want to be away from the water that's all out here. Okay, so the phospholipid bilayer, it's kind of like an important concept that we always have to learn about cells. Okay, so the plasma membrane, it's fluid, it's going to move, okay, it's not stiff and rigid. Okay, in it, it has proteins, and there's all these different kinds of proteins, so they can have different shapes and different functions, but all these like orange things are proteins. Okay, on the surface, we have some things like a glycoprotein, so that's a carbohydrate and a sugar put together. And to me, these are so interesting. They make up this layer that's called the glycocalyx. And the glycocalyx is all about identity. It's how we recognize self. So no two people, unless you have an identical twin, have an identical glycocalyx. So it's just interesting. You can learn more about it if you take more biology classes. Go to the end and record. Okay, so I'm just going to go. Okay, so structure of cells. So an example of a recognition protein that's found in the plasma membrane could be something like an insulin receptor. Okay, so you might have, if we go here, well, let me go back and then record. Okay, so a recognition protein, if I have a plasma membrane, okay, on that membrane, I could have like receptors in the membrane, and they can be receptors where certain things will bind. Okay, so for example, if someone has insulin receptors, which everybody does, so the receptors, the insulin can come up and can bind to the receptors and explain and allow the cells to take up glucose. I'll find a video and put in there to kind of explain that process. Another example of a receptor or a plasma protein that's found in a membrane is androgen insensitivity syndrome. So what this is that people that are XY, okay, they look like males, but if you have androgen insensitivity syndrome, you don't have receptors for the testosterone that you're making. So the testosterone can actually, I've spelled it wrong, the testosterone can actually work on your cells. So it's just interesting. These are just two examples. Okay, so now the idea, here's the nucleus. Okay, so the nucleus contains the chromosomes, which is the DNA. The chromosomes are, it's DNA intertwined with proteins. We'll talk about that. The DNA is important because it contains all the information for making proteins. Okay, and we'll talk about all the different things that proteins can do. Okay, so humans have 46 chromosomes. Okay, so 46 of these little, I always picture, when you say chromosomes or DNA, I always picture these little X's. Okay, so chromosomes are when the DNA is all condensed. It's wrapped around these little proteins, okay, and it ends up kind of looking like this. It doesn't look like this all the time in the cell, but it does when it's getting ready to divide. Okay, so here's the nucleus is what I want you to get. Okay, now, oops, what is going on? Oh, wow. Okay, so now ribosomes. Ribosomes are found in prokaryotes and eukaryotes. Okay, that's where protein synthesis occurs. Ribosomes can be in two different places, so they could be floating free in the cell, or they can be attached to the endoplasmic reticulum. Okay, if the proteins are made on the endoplasmic reticulum, the proteins will be transported within the cell in this endoplasmic reticulum membrane. Okay, main thing I want you to get is this. Okay, now the smooth endoplasmic reticulum doesn't have any ribosomes on it, so it's like here. It's all nice and smooth. Okay, so it makes lipids, it makes phospholipids, it makes steroids. Okay, so it's going to make other things. Okay, now the Golgi complex, what it can do, and I always think it looks like stacks of pancakes, so it kind of looks like this. Okay, it's going to modify and package the proteins. Okay, so it's going to take them and you can modify them. Okay, now this video, which I don't have it hyperlinked in this explain everything, but it shows you the process of how proteins are made and how they're shipped throughout the cell. Okay, now energy structures of the cell, so energy organelles, so there's the mitochondria. Okay, mitochondria are in plants and animals, and what they're going to do is this thing called cellular respiration. Okay, and cellular respiration is how we make chemical energy. Okay, how we make ATP, how we're going to take glucose and turn it into a molecule that we can use. So we're going to, ATP is the energy currency of the cell, and we'll talk about that more. Okay, this is a chloroplast. Okay, chloroplasts are found in plants and algae. Okay, what it does is they're going to take energy from the sun and make glucose. Okay, so here's more information about the chloroplast. So plants, algal cells, sun's energy, photosynthesis. I took a plant biochemistry class, and you can spend, like, there's whole chapters on this, so just want you to get the basic idea. Photosynthesis is this. Occurs chloroplasts. Chloroplasts are what make your plant cells green. Okay, so here's the mitochondria. Okay, so mitochondria are pretty much everywhere except for prokaryotes. Okay, so what they're going to do is they're going to take sugar and they're going to convert it to ATP. So the sugar comes from the plants. Okay, so you have to have oxygen for it to happen, and we're looking for ATP. Okay, because that's what we use for energy. Okay, now, chloroplasts and mitochondria, whoops, there's a typo, are what they call semi-autonomous organelles. So they have their own DNA and their own ribosomes. They have two membranes around them. They believe that they could have once been independent organisms, that they could have been bacteria that were engulfed by a host. Okay, and that's how they got another membrane. So that theory is called endosymbiosis. Okay, so sometimes scientists want to manipulate, like the mitochondria and the chloroplasts, to increase the nutritional value of the plant. So to try to maybe have the plant have more sugar, that it would be more, you could eat less of the same plant and get the same amount of energy. Okay, so think about some other examples of symbiosis. Okay, so a symbiotic relationship is when two things live together and they both benefit. Okay, so different cells have different amounts of the organelles. Okay, so for example, muscle cells have a lot of ribosomes and a lot of mitochondria because they need to make a lot of ATP to move, and they need a lot of protein because muscle cells are made out of protein. Liver cells have more vesicles that are called lysosomes because their function is to get rid of waste. Okay, now one of the big processes or kind of laws that we have in biology, that probably like the only one, we call it the central dogma of biology. Okay, and this will keep coming back over and over and over again. Okay, so the idea behind the central dogma is that if we need to take DNA and copy it, okay, that process is called replication. Okay, that occurs in the nucleus. Okay, now we don't let the DNA out of the nucleus. What we do is we make a copy of it. And when we make a copy of it, we call that transcription. Okay, so transcription is when we go from DNA to RNA. Okay, and that type of RNA is actually called mRNA for messenger RNA. Okay, now we let the copy, which is the mRNA, out of the nucleus. Okay, and from that, we're going to interpret it, and we're going to make proteins, and that process is called translation, and this occurs at the ribosome, okay, in the cytoplasm. Okay, I think that makes sense, but I do have a video. I have to figure out how to get those embedded in here instead of just being a file, and I haven't figured that out yet. Sorry. It says insert an object, so I could insert it, but I don't know how to do that. All right, we'll figure it out. I don't know how to copy. What's that? All right. Ah! Now I did it. Okay. All right. So this is just a picture that kind of shows what I just talked about. Okay, so here's the basic idea. Okay, so here's DNA in a chromosome, and it's unwound, kind of. And you can see the double helix. Okay, now genes are smaller and are within the chromosome. Okay, so you can have bunches of genes in one chromosome. Okay, when the time is right, what we're going to do is we're going to copy the DNA in order to get it out of the nucleus. Okay, and that process is called transcription. Okay, so we make the mRNA. The mRNA leaves the nucleus. Okay, it goes to the ribosome, and then from the ribosome we copy it, and that process is called translation. Okay, and translation is how we make proteins. Okay, so I always remember, okay, transcription, so that C comes before the L, so that you can kind of put it together that way. Right, so there's all sorts of different kinds of cells. Okay, and I kind of talked about that. So here's an example. These are Chinese hamster ovary cells, and they kind of, here's what they look like. Okay, so this is one kind of mammal cell that we use over and over again. We don't have to kill a hamster every time we want them. We can buy just the cells from a company. Okay, so they're used because they have a low number of chromosomes, okay, for a mammal, okay, but you still get the same kind of protein production as a mammal. They're very stable, okay, so they won't change over time, and we can make them express a lot of protein. Okay, so they call these CHO cells, so Chinese hamster ovary cells. All right, so types of cells used in biotech. Okay, so we have prokaryotic cells. Okay, so E. coli is the most common one. Okay, they're microscopic, you can't see them. Okay, and then we can use all sorts of eukaryotic cells. Okay, so there's protists, plants, animals, fungi. Okay, they contain membrane-bound organelles. Okay, so one example are HeLa cells, which have a very interesting story. I'll make another little assignment about those. Vero cells come from the kidneys of African green monkeys, and some cells that are used are yeast cells. Okay, so that's one part of chapter 2.